different backdrop than normal.
Ah, nice. How was your trip?
It was fun. It was very good. The weather was pretty nice, a lot warmer,
I
but
bet.
like not too warm. So it was still like wear shorts and a t-shirt, but also feel like comfy.
Mm-hmm. Good. I'm glad. Was it um kind of busy or I mean, it's always busy, but
It was like spring break time-ish. I don't think it was like the busiest week, but we planned pretty well. So we were like there were long lines, but we were kind of able to maximize our time, which was good.
good. I'm glad it worked out.
Yeah, very very fun, well needed break.
So back to the grind or not yet?
Sort of. Um, I'm traveling right now for March Madness, so going to all the like games that our basketball teams go to. So I might be grinding and traveling for a little while because I've got to
Gotcha.
write my thesis, but it's at least like a fun new location. So
Good. Where all do you travel to? Is it like all East Coast or do you move west too?
it's all over. Right now I'm in Columbus.
Okay.
Um, so that's where our women's team is playing for like the first round. And our men's team right now is in Brooklyn. So
Oh wow.
both
Okay.
kind of east-ish. But if
Mm-hmm.
we go on to the next round, those are in either Portland, like Oregon,
East.
or Dallas. So I don't
Okay.
even know where I'll be.
Yeah. How long does it last usually?
However long it takes, basically we go until the teams like lose.
That's I figured.
It's just kind of crazy because like you find out where you're going like the day
Day
that
of.
they win or lose. Yeah. So it's just a little hectic in terms of like knowing how much I'll be home versus not, but it's fun for sure.
When would finals be?
Actually don't have any finals this year, so it worked out well, but they're like I think last day of classes is like end of April. So
Oh,
um
I'm sorry. I meant like with the games, when are finals, like for the tournaments?
mm,
Sorry.
those are like the first weekend of April, I think like April 5th and 7th
Oh,
or something
geez. So
like
you'd
that.
be like out of school for potentially like a month too. Wow.
Yeah, we'll see.
Hopefully your professors and lecturers are all understanding about the situation because
Yeah. Well, when
I
you
don't
go to like a big basketball school, everyone understands, like, oh,
know.
I'm missing for basketball. They're like, oh, okay.
Valid. Sorry, I'm eating breakfast.
No, you're good. You're good. I'm sipping on my coffee.
Coffee is much needed.
Yeah.
How
I
big is the
did.
band? Sorry, I just want to ask one more
Oh,
question.
no, you're good. It's like 100 people.
Oh
When you
God.
travel for basketball games though, they only let you take 30 people. So it's like a smaller group.
So definitely the seniors. So they have the
Yeah,
experience. That's awesome.
mostly. hmm So I know like 2 years ago, our team went all the way to the Final Four. And so I had some friends at that time who were traveling. Like, I mean, they were basically gone all of March, but they had a blast because they went to California and then the finals were in like New Orleans or something.
Oh,
Um,
wow.
yeah. And they paid for everything. Like, we're in this like nice hotel, they fly us out. Like, it's definitely a
Um,
great experience.
I'm glad you're not driving because I would just be hectic of a road trip, but great for sightseeing.
Yeah. Yeah, that would be, I think it would be just a little too long to be on a bus with other people.
Oh, absolutely.
Mhm. Well, to change topics, I was gonna say I was looking over your like UWorld stuff, and it looks like you've been doing pretty well lately.
I know, it's insane.
I mean, it's good. It's a good
I'm
sign. It looks
taking it
like,
and I'm running with it. So,
yeah. I mean, hopefully this is like the hump that you've gotten over kind of thing. Yeah,
yeah.
it looks encouraging for sure. And I don't know, maybe just that like reset and then coming back to it. Hopefully that will keep your confidence boosted.
Yeah, I made a huge like in like in-depth slash dense list of how to organize myself for the next up until test day regarding like flashcards, questions, and then content review because I was snooping through Reddit and to see like what I should be doing regarding content the last couple weeks and also how it's going to compare from the practice exams once I do the AMC question packs and section banks. And apparently a lot of the feedback that I got was definitely to keep up with bio and biochem content and also psych and soc. So I went through more of Reddit to see what would be the most beneficial material. So it's more of not necessarily hardcore relearning, but just kind of the gloss over. And they said to they said that Jack Sparrow really helped like that deck. So I organized that based on foundational concepts. So roughly there are maybe like 150 per foundational concept. Some have more dense, like the metabolism and the organ systems, but I'm not going to touch the organ systems as much as the bio and biochem since I'm already kind of solid on those, aside for maybe just a few tweaks that I found through UWorld, which I already have flashcards about. But I'm just making sure that this time that I really do emphasize the missed content from the passages that I already seen like through UWorld at AMC. So that's where the flashcard base is coming from. And then the content review will be a mix of going through the foundational concepts for biochem, and then also incorporating Panko for psych and soc and a 300-page document so I can fill in those gaps. And then if needed, go back to Khan Academy for more of a video lecture based analyzation and then fill in the blanks with as much questions as I can. And then also like depending on the timing, how I am at the end of the day, I might even incorporate whatever I have from UWorld to apply those concepts that I drilled like that afternoon to passage. So then it will be easier for me to kind of like keep track and also apply those, you know what I mean? But
Yeah. Yeah.
do you think 180 questions is a lot? So that's 3 section tests per day. Because I don't
It's
want to
it's
also,
definitely pushing it. Yeah.
yeah.
My thing is like I think it's helpful to do like the half tests in terms of
Mhm.
timing and stuff, but then once you start to get into more like 3 sections, I think is where it starts to get like, the fatigue you have after you take a full length. Like I don't want that to happen every day kind of thing.
Right.
Um,
Okay.
so just kind of like probably would look at
No, but
to like what is it, 120 per day or so.
great. I don't know. It's just more of like how much am I going to have left before I start AMC, but I'm trying not to be discouraged by it because if anything, this is just going to help fill in gaps. And then AMC will be more of, okay, how am I going to really strategize the exam?
Hmm.
Um, but overall, I mean, from the practice exam, it showed well, like I felt good going through it. I had two notes. One is where do you have, like, as one source, have any like scientific notation practice, like to go through?
We might.
Because I just know that I spend so much time
Yes,
on
we
notation
do.
because I think it's just the nerves that just get to me. Because then after, when I go through it, I'm like, why didn't I just go through this blindly? Like it was fine. Because it's really straightforward, but then the nerves just get to me and then I overthink about conversions.
Okay. It looks like we do.
Because I know there's a unit conversion sheets that were already found on my what's it called? On my in my folder, but I don't know if that's the same thing.
This is like, this is a long one.
I mean, I don't care. I just need to like drill those again.
I don't know where the answers are, but I'll send you the first doc and then we'll see what happens if I can find the answer doc. This is like 42 pages, so you can just start
Okay.
wherever.
I'll take it.
Just do as many as you need to until you start to feel comfortable.
Mhm.
I'm gonna just email this to you.
Thank you. It's funny after going and reviewing AMC and then going back to UWorld and you're like, why is this so much more difficult? Like, why are you being so nitty gritty about some of the content?
Mm-hmm.
Just like analyzation. I'm like, oh my God.
Yeah. Yeah. And then like, you'll get this like random question where it'll have like expected you to know something and you're like, what the heck?
Mm-hmm.
Okay. Sent that. I'll look for the answer key because I think that'll be important.
Thank you.
But I can always just look over it. I don't know if I see an answer key.
No worries. This is great.
Uh-huh. Yeah.
Wonderful. Thank you.
Awesome. Yeah, hopefully that I feel like if you can just get kind of fast at that too, that's just like another helpful
Yeah,
like thing to have. Just like cut down on time, more efficient, that kind of thing.
exactly. Yeah. And I think that will really help to really just go over many problems and then it'll be easier for me to go
Hmm.
auto. I don't know what to do about
For
car.
sure. We can definitely think about it. I was thinking like we can glance through the car section that you did from this full length, and I have a couple ideas that I was thinking about, so we can just kind of go off of that. Um, Basically, just like maybe one or two new ways to kind of approach the questions themselves. Um, I don't know. It feels like, or I'm trying to look at what you've gotten right versus wrong. Like I think that at this point you've done a much better job of like getting a grasp of the passage and kind of what the passage is telling you, and it seems more question focused where the issues are coming in. Does that sound about right?
I think it's the opposite.
The opposite? Okay. It's more
I
passage.
at least from when I was gathering, where are you? From yesterday when I was going through. Um, I noticed there was a lot of more comprehension questions missed than usual. I also at the end, I don't know if you saw it, I like did it by passage on the topic, the score total, and then the skill. And
Hmm.
also on the left side of that passage, I wrote like the general feeling of what happened. Um,
Yeah.
and so the first passage was way too difficult, I thought personally, and I would not, it was just like whatever. Second passage was dumb mistakes. Third passage was history. I ran out of time, but I managed to go through two of the seven questions, and those two were right anyway. Theater based was, I missed, excuse me, the main idea completely. And then
Uh-huh.
religion was the last one. It was easy, but my comprehension was bad. So I think it was the way I was, I mean, I might be over highlighting, honestly, but for what I think that I might need to highlight might be a little too much. So I started pinpointing about maybe I should just highlight what I think is the main idea, the evidence supporting the main idea, and then the examples that support the evidence. Because it's all really asking about to take one of those three things and to either apply it or apply it to the next level. And
Hmm.
if I can mark those, I might be okay. Because the questions make sense to me during, I mean, nerves are going to get to me, whatever. But I think if it's also lack of confidence and self-doubt from understanding the passage, then the answers are just going to follow with. Um, but for the most of it, I understand the passage. I think it's just really about those nitty-gritty details about what the author or the AMC writers want us to remember or at least be more mindful about versus the other details, if that makes sense.
Hmm.
So
Okay.
again, that would probably be like one or two highlight for each, but also keep in line of what is the difference between like subject A and subject B, because like that was also part of like a good portion of what they do for cards as well.
Yeah. Okay. I wonder then, so I know we've obviously talked about the highlighting strategy, and I do like it. Kind of as like a supplementary strategy to that, something I sometimes like is almost kind of like diagramming the passage out, which can take a bit of time, but I think if you are just very shorthanded about it and just use like symbols, it can be a lot more um, direct. And I mean that in like, so if you've highlighted the first sentence, you kind of know that it's going to be like, first sentence is your like thesis of that paragraph, and then evidence, evidence, evidence kind of thing. Just to kind of like map out like, oh, if I need to look for evidence of this, I'm going to like look here. And then that can kind of
Oh,
almost
I see. Okay.
also highlight. I'm going to try to show this on my, I'll draw it out because that will make a little bit more sense.
You didn't bring your
Um,
whole pad with you, did you?
no, I just
Oh,
brought my iPad.
okay, good.
That
I was
thing's
being like,
like,
that's really dedication, man.
oh my God. Yeah. I mean, like, I could probably fit it in my suitcase, but
Oh my
I don't want
God.
to risk that. That would be a little
TSA would be
intense.
all up in your business, being like, what do you need this?
Yeah. Although we do get to fly on, like, we fly on like a charter plane, so
Okay.
they don't like actually check my luggage. So just like roll up. Do you think it would be a bit excessive though to show up to my hotel with like a whole screen? Um, trying to invite myself to the Zoom meeting. Hello.
All right, I'm just gonna get some tissues real quick.
Oh, no worries. I will try to figure this out.
It's snowing again, or it did snow earlier, so it's like been screwing up with my allergies and my headaches. I'm just like, can we
Oh,
actually have spring, please?
yeah, that's rough. I was just gonna say I my allergies have flared back up because I went to Florida and it was warm, and there was also a big warm spell, but now it's getting cold again. So my allergies are just all over the place.
I love it.
Yeah, we're staying alive.
Yeah.
Okay, I think it's letting me join. It just says waiting. Error. Oh, I need to connect to Wi-Fi. That would do it. Okay, we're getting there.
You're
Stream.
so fun.
No. Okay. The other thing I was looking through that I'll say, and we can get back to that, is the previous like exams and what those scores were versus the scores when you took it again this time around. I don't know if you kept track of that at all.
Mhm.
Um, but it definitely looks like there was some pretty decent improvement.
Yeah,
That's
four
likely,
or
hmm?
like 4 points in each section aside from cars.
Yeah, I think that's awesome. I feel like generally with retakes, it's like if it's solely due to the retake, it's like max 2 points. So I don't know. I think that could be a very good sign.
Yeah,
Because I don't think it's just retake. I think it's also a better kind of grasp of things because it's also correlating to the UWorld going better.
I agree. I mean, your world has been like the average has been increasing with which has been very comforting. I'm like being really chill about chem and physics because I think that's just going to be, if it's going to improve, that's just going to be from AMC strict material and reviewing. And then the last two sections are mixed with content and applying and reading. But then cars, I don't know what the hell was going on. If that's just a passage thing or I don't know. Because like I don't know if you've seen also like the cars I've been doing with diagnostic, they've been fine. And my timing has been decent too.
Mhm.
So I have no
I
idea.
mean, to the cars, I think in the later sections, like the later tests, so like 3, 4, and the scored,
Yeah,
I think are the more because they're the more recent ones, so they have the more like up to date language,
right.
style of logic, and stuff. I always am hesitant with like the first AAMC exam just because I know that one's a bit older. It gives me the same vibes as like the first half of Cars QPAC 1 kind of thing.
I know what you mean.
Um,
And we also ended up choosing the one that had the 3-7 questions, which was
yeah,
also
and we had
I
said that was weird.
yeah. But on the like the funny part is that two of those three ended up doing well,
Mm. Well, I
which
think
is
that was
ironic because like the sociology was 4 out of 7, philosophy was 5 or 6 out of 7, and
yeah.
then art history was a 3 out of 5. So I was like, you know what? Okay, I'll run with it. It's fine. And then ends up screwing up the other 5 questions. It's fine.
Did you at all compare like what you got right the first time versus right the second time? Um,
Um, I did with biochem and cars. I remember going through psych and soc and biochem and being like, I made the same mistake, or it was like the same
mhm.
questions that I got wrong. And the worst part is that I even wrote like a flashcard and I didn't get to it previously. So it was more of, well, if I would have gotten to it, would I have done it or approached it differently? But I was too scared to see what was the comparison between cars. But I know that I had less yellow on this round versus last time, if that like the run out of time. Oh no, you froze. Hello. Is it my Wi-Fi or yours? Oh, I think you froze.
Okay, there we go. It like exited me out of it.
Oh,
Weird.
I don't know how
Okay.
much you heard. So,
No, the last part I heard was that you were going through comparing like when you made the same mistakes, and then you were like, the worst part is, and then I never
oh,
heard what the worst part was.
I just didn't really want to go through cars because I know that cars was going to be a disaster. But what I know and caught between the two different car sections was I only ran out of time on one passage versus like two or three.
Hmm.
So I think timing has improved.
Hmm.
I don't know
And I
about the,
know when you did this passage the first time, that was when you also had like run out of time but kept going, right?
I think so. Yeah.
So that kind of score from the beginning was also like wouldn't have necessarily quite been at the 122 just because um just be
It really doesn't like your iPad.
Ah, why did you disconnect? Or my headphones are being weird now.
Can you hear me?
Yes.
Okay, because I don't think you're through your headphones.
okay.
Technology
But
is finest.
it wanted to like go through my phone for some reason.
That's what it does with me sometimes. I don't know how to stop it.
I don't know. It must be the new like update because I just updated to like iOS whatever.
Mhm.
Okay. Anyways, where was I? Oh yes, the first time you did that passage or the first time you did that section, you had run more out of time. So even just on like a pure timing level, you've definitely improved just from like the first time to the second time, which is good.
Yep, I'm pretty pleased with that actually too. So
Yeah. I think overall, like things that are like just easy to fix by repetition are starting to get fixed. And now it's just honing in on the topics that are going to be more like you have to actually do the review and, figuring out the right question strategy and all of that.
yeah, and I think a lot of that will also help regarding um section banks and question packs once I get to it.
Mm-hmm. Okay. I was just going to sketch out this idea really quickly. Kind of like when you're reading the passage, you kind of have this like structure where it's like introduction to idea, would be like the first paragraph. The second paragraph, you kind of get like maybe it's like
Some background, yeah.
background, da 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 da, and then the last sentence of that is like your thesis, your main idea, kind of thing, and that's what you've ended up highlighting. But sometimes it can just be helpful to see like, okay, this paragraph is kind of set up as like thesis, supporting evidence, like one, like counter argument, and like explanation to the argument. Just to kind of look at like what the structure of the paragraph is. So when you're looking at like, oh, someone disagrees with this, you kind of know you've got like, here's the idea they disagree with, and then here's how the author like responds to that, something like that. Just thinking more about the overall like actual structure of the passage, not even necessarily like the content, but like what it how it's kind of mapped out this argument. And that's where you can also just pull aside like counter argument, like Bob thinks XYZ, blah blah
Mhm.
blah. Um, and so that is where you could just highlight like these are not author ideas, but they are mentioned in the passage. Because you'll get those kinds of questions like what would the author think, but they've included something that's like the counter argument to the author
Mhm.
versus something more down here, author ideas. It's going to look directly at like the counter arguments, your evidence, etc.
Mhm.
I know this can be fairly like light in terms of actually like what you write down versus map, but just to kind of like make sure you have a good grasp of the structure. And then I feel like what you'd want to write down more would be like on this side of things, the what the author's thinking versus what's the opposing viewpoint, just to have like those as like big bullet points. I feel like usually it's like what the author thinks is like everything that's highlighted, and you can just kind of like note that. And then what you might not highlight or might not draw necessarily as a main idea would be like over here where you're potentially thinking about the counter arguments or something that maybe the highlighted ideas respond to,
Mhm.
something like that. I think it can be helpful to almost just like column A, column B, like here's what the author think, here's what other people think.
That's what I was
Um,
thinking, like a flow chart, kind of more or
mhm,
less.
yeah. So it can almost look like, I don't know, do you remember there's like a Cars QPAC passage about gossip? You might not have gotten to that one yet. I'm trying to think of what's one that's early on. Okay, I'm just gonna make something up. Okay, so you get this like thesis idea. Animals are cool. You're kind of like going into evidence 1, which is the first thing you've highlighted in paragraph 3, you've got like those sub explanations. You've also got like this branch off over here that it's like, I don't know, Joe disagrees. I think that kind of referencing sort of that flowchart idea that you had. And then why does Joe disagree? Here's his evidence. If there's now evidence that connects it back that the author is using as like a justification, whatever that will look like. I don't know if you're more like, if you're generally a visual learner. I just learned this term like kinetic learner, where if you write stuff down, you're like learning it. I've just realized that's what I am, but whatever kind of works to just flesh that out and to also have just like a couple points here that will help you differentiate like opposing viewpoints or slightly different viewpoints.
Yeah.
Okay.
I think today is going to be a lot of that experimentation of how to write it out and
Yeah.
see what would make sense and like how to make it as efficient and not as time consuming.
Yeah. And like, and it's hard to give you like exact details, but I think that what like you'll find something that works specifically for you.
Right.
Because it's very like person to person.
Right.
I would potentially like just mess around with like the passages you've already read that you're like pretty familiar with. Yeah. So you could even do it with the ones from this full length.
Yeah.
Okay. I wanted to jump into the UWorld questions that you had from last week.
Yeah.
Yeah, these were the bio is looking really solid, which I love. And then it was this guy. Yeah. Okay. So I wanted to just kind of like briefly run through the passage, but these seem mostly like genetics-y related. And then just think about some of the couple of shortcuts that might exist, but um, we'll just kind of glance through this again first. First of all, just to comment kind of on your highlights. I mean, fairly straightforward. Introducing, basically, we know fruit flies will mirror what regular humans look like. Introduced to 3 genes. Okay, believed to be in the same pathway. I like that you highlight this. Fly lines with dominant mutant versions of each gene were developed. So we know that what the dominant mutation is, and it's going to be denoted by a uppercase, and then we can also like just see what that phenotype would look like. This is where we just get across. We're probably just going to have to draw that out. We know these guys are located on the same chromosome, and then we're doing recombination and looking at the number of flies. Okay. Overall, not like a super detailed passage. I think it's definitely going to relate more to like actually doing genetic crosses and that kind of thing.
Yeah. The passage seemed straightforward. The questions were really just out.
Okay, so we've got picture of homologous chromosomes. The diagram above represents the immature gamete of a male fly that is heterozygous for Ina. The gamete undergoes meiotic division, and Ina alleles do not undergo recombination, during which phase of meiosis will the sister chromatids containing the little e allele be pulled to opposite poles of the cell. So this is very much just kind of checking in to see what you know about meiosis question. And since there's no crossover, we know that it'll just kind of like pull each strand apart as is. So don't necessarily need to worry about that. Okay. Do you remember, I mean, with meiosis, obviously there's like 1 and 2? Like, do you remember kind of generally the stages of what that goes through?
Um, more or less, yes. Like from prophase 1 to the end.
Mm-hmm.
Okay.
Yeah. I mean, the nice thing about my meiosis is it's very like based off of mitosis. You've just got to remember some small details.
Right.
Uh,
I think it's even anaphase that is the most different out of the two. It's just more of which one contains what.
mhm. Because prophase is pretty much the same in each, like prophase 1, prophase 2 are like the same-ish thing. It's just like what chromosomes are actually there. Metaphase 1, also same idea, is just like they're lining up along the center.
Yep.
The only difference I would say is in metaphase 1, and this will kind of make sense too, you get this lineup looking more like this
Right.
versus in metaphase 2, it's more like this, and that just has to do with the number of actual chromosomes that are there. So that when you do pull apart, it kind of makes sense that you're cleaving off this half and bringing it this way, and then this half and bringing it this way. Versus for this one, you're splitting it down the middle.
Right.
But same idea, they're lining up on the plate. So yeah, anaphase is really where the difference is. And that's just, you can think of anaphase 1 as just like separating your X's. There's no like breaking up of centromere yet. There's no like actual pulling apart sister chromatids. You're just kind of separating out your X's. And then in anaphase 2 is where you're actually going to start to pull apart the sister chromatids. And you can think of that step as like, this is where you're actually starting to like break apart a structure.
Mhm.
Before you were just separating out, like just kind of like sieving out different X's and like one goes this way, one goes that way kind of thing.
Right.
Now is when you're actually pulling apart the different X's chromosomes, and pulling apart the sister chromatids. So I don't know if there's like a way to remember that particularly, but I just kind of think of anaphase 1 is like just, more just sorting, if that's helpful. And then anaphase 2 is when you actually get the like pulling apart. Does that make sense?
Yes.
Awesome. Yeah, I think that one pretty much just depends on knowledge of the anaphase 1 versus anaphase 2 kind of thing.
I think it was the do not undergo recombination because I knew it wasn't metaphase, period, but I couldn't
Mhm.
recall if it was 1 or 2 because I knew the one is that one of the two also goes through non-disjunction as
Yeah,
well.
so that's gonna, let's see if
That's
I
anaphase
should have
1.
it in the picture down here. Yeah. That's gonna be over here, but it'll still be like most of the one chromosome. Because you can imagine in this little picture right here, like there's kind of this, if they've crossed over, there's like this portion and then like that portion. I don't know if you can even see that. That are like crossed over, and then this portion crossed over there, and that portion crossed over there. So like when you are pulling apart, you are technically like pulling apart and breaking that like bridge that had formed between the two, but as a whole, it's still can kind of think of it as like separating an X from another X.
Mhm. So crossing over did happen in anaphase 1.
So crossing over would happen like before anaphase 1,
Okay.
but that's when you would pull them apart and there would be like, you would get your final result of like this chromosome would look like that, and then
Mhm.
this chromosome would look like that kind of thing.
Right.
So that's kind of where you would see like, oh, these are what my new like recombinant chromosomes actually look like.
Mhm.
Awesome. The crossing over itself actually happens, I'm pretty sure, during the prophase phase when they're just kind of vibing.
Okay,
And then they will separate and undergo their normal thing.
got it.
Okay. Cool, cool. Let's kind of clear this. Okay. Ah. Okay. This one was just pretty much make a Punnett square type thing. Were you able to get this pretty straightforwardly?
I guess. I just knew it was going to be less than half based on the passage, but I knew it wasn't going to be as small as 1/8. But
Mm-hmm.
I don't know how to do the 4x4 Punnett square scares me. So I don't know. I don't know.
Yeah, that's fair. Okay, let's look at so female EEPP were crossed with males EEPP were then selected and crossed with each other to produce an F2 generation. Okay, so basically we're looking at big E, big P, little p, and then crossing them with each other. So the first thing that I like to do in this kind of scenario is just kind of break apart what is actually happening here. So this is the phenotype that we're working with, or the genotype. We know that when we get our gametes that are going to have a single pair, there's only a certain number of combinations that could happen.
Mm-hmm.
We could get it looking like this: EP, 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 or EP. And that's basically like which one of the two did it pick in each of these cases.
Mm-hmm.
So I first kind of like to break it down to like, here are the 4 scenarios, and it's always 4 scenarios or less, depending on like if these were both big P, it wouldn't really matter. You would only really want to just cross the E's to actually see what was going on here.
Mm-hmm.
So first you break it down into the scenarios. So if you have 2 genes, it's a maximum of 4 different things that are happening.
Mm-hmm.
And then I'll also write that now going in this direction. There, let me do them in the same order so that makes sense. Okay. And then I'll just kind of draw lines in between each of these. And you essentially know that when you're doing 4x4, you're left with 16. That sounds about right, right?
Mm-hmm.
And so you know that whatever you're going to do is going to be some fraction out of 16. So, I mean, if there were any of these that weren't like,
Divided by 16.
like,
Divided.
then you would know they were wrong, but they're all potentially right because 8, 4, 16. Anyways. And then you want to figure out, we can write out this whole Punnett square chart if you want, or we can try to find out maybe a shortcut. We're looking for the ones that do not survive, right?
Mm-hmm.
You know, that flies with double E and at least one, and then this E or this do not survive past larval stage.
Mm-hmm.
So the first thing we can do is just look for where does the double E cross over. So we're looking at this one, this one, this one, and this one, and any squares where those cross over, I'm just going to shade. Because we know all 4 of these would probably die, right?
Yep.
They each would have a big E. So that takes care of this side as well as this one, because any ones that have EEPP are also going to just have EE. So we've already accounted for all of those. And so now we're going to just look for big E, little e, PP. So we already know that if it's going to be P's, it's going to have to be within this row and this row,
Mhm.
or this row,
Mhm.
and this row. So here's our two. It would be somewhere in this like first two rows of each. That's where I will potentially just write out the genotypes here. This would look like E, little e, PP. This would look like E, little e, PP. This would look like little e, PP. And of these guys,
Two of them.
these two would end up being lethal. And those
Okay.
are all the circumstances. And at that point, we've just got 1, 2, 3, 4, 5, 6 out of 16, or 3 out of 8.
Got it. Okay.
Does that feel more efficient than actually trying to write out the whole thing?
Yes.
Because I think that's just a better way to like show, like without actually having to write it out, just kind of shade it in. We don't actually care what the whole genotype is. If they have 2 big E's, we know they're going to die, kind of thing.
Mhm.
I think that's just a little bit easier than trying to write out the whole square and then look for all of those, but maybe just kind of shade. And if you need to test out what the actual genotype will look like, you can draw it out from there.
There was like something that I saw at one point that was like a shortcut of the ratio with this kind of crossing that it's like a 9-3 to 3-1.
I think I know what you're talking about. I'm gonna erase this.
Six, that's 16, 9, 3, 3, 1.
Square, where are you go? Square ratios, dihybrid. It's always hard though, because that's only going to specifically apply if you have this exact genotype where it's heterozygous for each gene. Um, so that's what that shortcut's for, which is what the situation was, where it was like big little times big little. However, if you were asked a question in terms of like what was the original cross, which was these guys,
Mhm.
like little big little, or big little, then that ratio wouldn't apply. You could definitely memorize the ratio like if you wanted. Here we go. But I think that it's at least easier for me to just kind of sketch it out and do the shading, than try to think about it in terms of the ratio. Just because
Yeah,
I think that,
I never
here we go.
understood what like the ratios meant either. So if it's not worth
Yeah.
going through that, then it's not. Yeah.
Yeah. The ratios are going to be like if one is dominant and one is recessive, and you have 2 heterozygotes for each gene crossed with each other. So it's like a very specific set of circumstances. Whereas, if there was some other different type of circumstances, if you're crossing any other genotype, it just gets a little harder. I think if you're just able to draw out like just the edge of this guy and then the edge of this guy and then do your shading, kind of like how they've done it here, but without even writing anything in, I think that's generally for me the most efficient way.
Okay. As long as you pair it correctly of that first like row, not just saying it's big E, big P, little p, but actually like with itself. Like the big E, big E, little p, big P.
Mm-hmm. Well, it doesn't matter what order you write like these out in.
Right, but I'm
It'll
saying
like that'll just change.
having that combination, not just singular E, E, P, P.
Yeah, I think I get what you're saying. Is it like just making sure that you're getting the right rows kind of thing?
More or less, yeah.
Mm-hmm.
Because you're crossing over 2 and not 1. Or crossing Yeah, you're crossing 2 with 2 and not 1 with 1.
Yeah. Because what it told us and how I had done this was like, this is lethal. And then we also knew that this was lethal. So these were the two things we were looking for.
Mhm.
And how I had identified that was like, I know it's got to be somewhere in this row and then in this row, and basically just looking where the two E's overlapped with each other,
And
which I
then
think should kind of make sense.
the big piece, which is last, which takes care of the other two.
Big P's, which are, yeah, it's like, you know, that the ones you're looking at have to be somewhere like in this crossover space. Okay, makes sense.
Yep.
Awesome. Yeah, I think that's shading's the easiest way to do it. Okay, of the genes tested in the passage, which are autosomal?
LOL, this is we talked about this before.
I think so. All believed to operate on the same pathway. I don't know if it necessarily says anything directly in the passage, but I think it's like the implied one. Yeah, it doesn't necessarily. Oh, I think it's from. Let's see. Males in experiment 1 were heterozygous for ENA, suggesting that they had 2 different copies of the gene. Therefore, ENA cannot be on the X chromosome and must be autosomal. So that's why I was saying like ENA had to be autosomal, which makes sense because that's where we did these crosses. So they would probably have to be like autosomal crosses in order for us to actually get the results we got from the last question. And then this would basically just be thinking about like if it was sex linked, there should be kind of some differences in male versus female offspring, but we don't necessarily see that. This line right here produced an F2 generation in which 50% of both male and female progeny had elongated eyes. If it's something's affecting males and females equally, it's generally going to be autosomal rather than sex linked. This is like a random, because the passage didn't even differentiate between males and female as this like final result, then it's probably all autosomal linked.
Okay. So so what did it mean by same proportions? Is that what you mean by like there's not a differentiate between males and females?
Yes. So if there was some kind of sex link going on here, you would see a difference between male and female offspring, right? So this sentence here that says an F2 generation in which 50% of both male and female progeny had elongated eyes tells me that males and females have the same outcome, so it's therefore autosomal linked. And you have a big enough like population size, this is like what, like 500 flies, that there's no like by chance you would definitely have to see some kind of difference if there was a sex linked component going on here. So not even like mapping out what's happening or like thinking about it, but just saying that because males and females both have the exact same like phenotypic ratios, it's not sex linked.
Okay.
And like I'm not even thinking into it much further than that. And this is talking about Able, and it tells us that their PTP and Able are on the same chromosome. So that's how we get that PTP and Able are both on the same chromosome, and so they both have to be autosomal. And then from ENA, that's where we got that that one would also have to be autosomal because we saw that males had 2 copies of a gene, which they wouldn't have if it was X-linked.
Got it. Okay. Because then for experiment 2, if it were sex linked, they would be on different chromosomes, right? Or
Yeah,
not?
if it was sex linked and they were both on like the X chromosome, then you would have to see like they would have to differentiate like, oh, 100% of female prodigy had like regular eyes, and then like
50.
75% of males had this, 50% of males had this kind of thing.
Got it. Okay.
So I think it's like generally just like do they differentiate that something different happened between males and females? If yes, then you take an extra second to double check and make sure it's probably going to be sex linked. And if no, then it's pretty much for sure autosomal.
Okay.
And that's just like a very like high level sanity check, probably autosomal. Which evidence in Table 1 suggests that PTP and ABL are located close together on the chromosome? Okay. So I'm going to just start to sketch out what this would look like. So we're going to imagine that this is my chromosome. So I'm going to say this guy is PTP, and then this guy is ABL. So generally, when you're thinking about these kinds of questions, they're asking you to think about recombination, which we talked about slightly earlier, the idea of like crossing over, right? That's how you're getting all the genetic diversity.
Mhm.
And the simplest idea is if they're closer together, it's likely that they'll be like cleaved off together and then transferred to the same like chromosome together. Versus if they're further apart, there's more of a likelihood that they could be cleaved up in between them, and one allele would stay on the original chromosome and one allele would cross over with the other chromosome. So basically, recombination between the two genes, the closer they are, the more they stick together, if that makes sense. The closer they are, the more they stick together. The further apart they are, the more likely there's some recombination and one of the two gets separated from the other. So I'm going to kind of draw it out with colors because I think that's probably the easiest way rather than like big P, little p kind of thing. Okay. So if we're crossing heterozygotes, which I believe is what's happening here.
Yeah.
Yes. We're going to imagine that blue is mom, green is dad. And these are, blue is one type of mutation, green is the other type of mutation. If there's no recombination that occurs, one gamete is going to get all blue, and then the other gamete is going to get all green. And that should make sense, right?
Yeah.
If recombination does occur, there's several kind of things that could happen. One, maybe the crossover point is right here. So this part of this chromosome becomes green, and this part of this chromosome becomes blue. But PTP and ABL are both together. There's still one blue PTP and one blue ABL in this one, and one green PTP and one green ABL in this one. Makes sense. However, recombination could also happen, and let's say the crossing over point is right here. Oops, that's not what I wanted to do. Right here. So now we've got green PTP with blue ABL and blue PTP with green ABL. So now this is a phenotype that did not exist in either of the parents, but now exists in the gamete, where you've got a mix of green and blue in each of these. So that's where recombination can create genetic diversity, because now this is a phenotype that was neither seen in the mom nor the dad, but the child can have this if they come from this gamete. Essentially, what this is asking is, how do we know that they're close together? And the idea is, the closer they are together, the more likely they are to stick together. You know, if the distance between them is really small, the chance that one of those like crossovers cleaves right through the middle of that is also very small. So the closer they are together, the more they stick together, and they're going to be stay blue with blue, green with green. The further apart they are, the more likely that when they cross over, they get separated, and that's when you get your blue green, blue green.
The difference. Okay.
So basically, what we're looking at here is parental versus recombinant. So in this case, parental, we'll call this one. This would be like our blue scenario. This would be like our green scenario. This is where both are green or both are blue. And then this would be your situation where you would get,
Some
okay,
blue,
here
some
there was some
green.
kind of like, yeah, some blue, some green, and then some green, some blue. And you basically just want to look at the frequency in which that happens. So how many recombinants are there compared to parentals?
But we're comparing
If,
to the total number of flies.
yes.
Okay.
So if these are on separate chromosomes and completely unlinked, you would generally see about 50-50. That would be like your normal, like there's a 50% chance someone gets green, and then there's a 50% chance someone gets blue. So you would see both an equal mix of kind of recombinant and parental cells.
Oh, okay.
Almost in terms of like, if you're crossing, that would look like this. Let's see. Green, green, times blue, blue. I don't know if that exactly works, because they're different genes.
It still work.
Oh, well, it would look like this, actually. It would be blue, blue, green, green, because each parent cell has one blue and one green. So you'd get 50% of your progeny are going to be blue, blue. 25% are going to be blue, blue. Sorry, 25% are going to be green, green, and then another 50% are going to be the mixed, which would be these guys. So if they're totally separate from each other, not close at all, recombination, like just by random genetic chance, there's going to be 50% that are recombinant and have a different genotype than their parents. And that's just like your normal Punnett square. And that's what we see happen when we cross, like, you know, normal things for genes that are unrelated.
Got it.
If they're closer and closer together, it's not just a simple question of like, one's going to get blue, one's going to get green. The two blues are going to stick together. The two greens are going to like to stick together just because they're so close. That's where this question is asking. And so it's asking us to just look at this number. If the recombinant number was a total close to like 250 out of 500, that's where you would say like they're probably pretty unrelated. They're just like on different chromosomes. They're not close to each other. They got recombined. But if there's more and more that only have the parental phenotypes, that's what indicates to you that they're closer to each other. They stuck together this whole time. They did not get, if they got recombined, they got like recombined together. They did not get like separated down the middle, because they're so close that there's just such a small number of scenarios in which they actually do get separated from each other.
So then what would be the opposite if recumbent was taken over versus the parental? Would there be more recumbent flies versus the parental?
You would see it as like there would be like half recombinant flies and half parental flies.
Okay.
So each would probably end up like it would almost look like a quarter, like each genotype has about equal frequency of occurring. So like they would all have like 125 flies or so.
And what did you say that if they were far, what would the numbers be? What would be switched?
Yeah. So that would be what I was kind of saying with the 125. If they were far apart, it would be like equally as likely that you get a recombinant versus a parental. And so that's where you would just kind of see like the same chance of getting any gene.
Interesting. Okay. I mean, it makes sense then. But yeah.
Yeah. I'll just also kind of redraw this as our larger picture. This is kind of what you'll see. It's a question of if our genes are here and here, you can imagine that when there's crossing over occurring, this whole leg is kind of being crossed over together. So like
Yeah.
these two genes are sticking together versus if our genes are here and here, there's a much larger chance that like this portion gets crossed over and switched with green while this portion stays here. So that's why in this case, you would see lots of recombinant. This would be the case in which it's like just pure chance of whether you get recombinant or parental. They act kind of as separate genes. Versus if they're close together, that's why you see more parental because they're not actually being switched out with each other. They're traveling together during the crossing over event.
Okay.
Does that make sense? I don't know if I've explained that
A little
as
bit,
well.
but clearer than before, for sure.
Mm-hmm. Basically, recombination frequency is low when they are close together. See if they've got a picture here. I don't think they do. Not a good one. I can try to like explain it slightly differently if you want to hear it in a different way, if you think that would be helpful.
I think what would be best if I just applied this to something like a different, like something else. I don't know. It's like I
Mm-hmm.
understand how recombination works and all of that. I think it's just more of like seeing it in data and
Yeah.
the application process of it. But overall, it makes sense.
Mm-hmm. Yeah. Okay. Cool. Basically what I was this was what I was just drawing out before of like if your two genes are here and here versus if your two genes would be here and here, it would depend on whether or not they stay together, if they're close, or they separate from each other if they're far apart, kind of thing.
Yeah. And then the recombination frequency would be low if they are close.
Correct. This is big P. Well, P is not a good letter. It was hard to tell capital. This is big B, and this is little b. Or if this is big B, and this is big E, you would see like the parental phenotype is BE. If they're far apart, you'll get a mix of like, let me draw this. This is going to be little b. This is going to be little e. It kind of makes sense. These are your two parental phenotypes, right? Red has little, blue has big. If they're close together, you'll only see like a lot of each of these because big B and big E would stay together, or little b and little e would stay together. Versus if they're far apart, you can see where in this case, you'd have big B little e,
Mm,
and little b, big E. And so
mm,
these are the recombinant chromosomes that now have new genotypes different from their parents. And so the more frequent these are, the further apart they are, because this crossover kind of interceded in the middle of them. Okay, I'll leave that there. Hopefully that makes more sense in future applications. I feel like if you do get a question about it, it's pretty much going to be this type of question, where it's basically asking you to look at the number of recombinant versus the number of parental. So if you can just understand that low recombinant means close together, then I think you've pretty much gotten that.
okay.
Okay. So we have a genotype of a female was unintentionally mated with a single F1 male of an unknown genotype. If the 25% of the resulting progeny died, the genotype of the F1 male was most likely.
Backwards.
Because this is like where it would be helpful to have that like whole Punnett square thing, but we also don't know what the one side of the Punnett square would look like. Okay, so we knew from before that EE is lethal, right? And then
Mhm.
E little e PP is lethal.
Mhm.
Okay. And we know the mom is going to have EP, 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 right?
Mhm. And 25% died, so that's 4 of the 16 is gone.
Okay, so we know for sure it's got to have to do with these first two columns, because there has to be, or there's, well, it could potentially relate to the other columns. What I'm seeing is we know that if it has big E, we know it'll die. I'm probably focusing on these two columns. I think the easiest way for me to do this would be to input the male genotype and just see if I can scribble out 4. And then if I see that it's more or less, then just eliminate that as an answer choice. My first thought is looking at B and C, since they both have
Little e's.
like little e's, those are probably going to be a lot less likely,
That's what I
because we
thought.
know that in each of these cases, they would have a little e no matter what.
Mhm.
And especially with C, like the only thing they can possibly get is little e, little p. So that immediately takes like all of this off the table. And yeah, pretty much prevents you from even having a double big P phenotype or a double big E phenotype.
Right.
So pretty sure it's not C, just because we can't ever get a double dominant mutation.
Right.
So I'm going to take C off the table. Okay. And then we know B would all have to look like this. So again, we can pretty much eliminate this, because all of these, well, we can eliminate all of the little e ones, right? Can't be any of these. We could just kind of draw out what these would look like. So this guy would die. This guy would be alive. And so we already know it's not this one, because I think only 2 of these guys would die, and it would be these 2. Because only these 2 can have double P's. I think this is the most efficient way to do it.
Okay.
If you try, because I'm thinking like if you tried to do it like any other way, like mapping out a whole Punnett square, like you'd pretty much be doing the same thing, but this way is just avoiding actually having to write out every single like genotype.
Mm-hmm.
Okay. So then let's look at your choice. This is where we actually have some different things. We can have E big P, E big P, little e, big P, little e, big P. Again, we don't care about all these ones with two little e's. We can kind of just draw out.
Everything. All of them would die.
P, P. Which would be 4 out of 16. Making sure I did this right. Sorry, I'm just looking at this. I'm like, this would kind of make sense. This was stress D.
They're absent.
Oh. So this would make sense in a genetic sense. However, we just said that big E, little e, big P was lethal. So it's basically saying like these flies couldn't exist, let alone reproduce.
Wow.
That's tricky.
Okay.
So it works in a Punnett square sense. So you did get that right. Like you had the right reasoning here. However, if they're dead, they can't reproduce, can they?
Okay.
That's tricky. That's
Valid.
mean.
That's
Okay.
valid. That's so valid. But rude.
Yeah. Well, and I guess in that sense, it was easy to eliminate B and C, can also then be easy to eliminate D. Maybe you don't even have to sketch anything out and you can pick A.
Precisely. Yeah, that's now what I'm thinking. Is this just more of a let's see if you can reason your way through the choices rather than go complicated.
Mm-hmm.
That's
But hopefully the scribbling out method was still helpful. I think
yeah.
I think it's a better way to approach. I don't know. I just can't, if I see too many letters, it like hurts my brain. So
I know that's
I
why
think
I
as
was like,
long
I'm just gonna skip.
Mhm. Okay. So we now know, pay attention and don't pick any dead flies. And then for the rest, just quickly sketch it out. If there's one, like for this one, for these ones, you can kind of like gut check them, but you should pretty much be sure that these would not work. Kind of thing. Cool. Okay. You're tricky. Okay. Looks like
Missed this
you
one
did
big time.
not answer this one. Okay. Axonal guidance is the process through which axons reach their proper targets in the brain. Researchers hypothesize that the wild-type PTP gene product mediates proper axonal guidance by inactivating the ABL gene product. So PTP, ABL. Which experimental observation would best support this hypothesis? A, suppression of a, okay. I think the best way to do this would just be to look at like experimentally wise what would prove this connection.
Mm-hmm.
So it's saying that like basically high PTP would mean low able activity.
Which means low axonal guidance.
Which means well mediates proper axonal guidance. I think it would mean good axonal
Yeah, you're right. It would be high axonal guidance means
because
low
yeah, because
ABL.
it just says like mediates proper axonal guidance. So we want a case that so high PTP means low able means high axonal guidance. So we're potentially looking at a place where like if you have high able, you have bad axonal guidance because PTP is not acting. Okay. So let's look at D. Overexpression of able restores normal axonal guidance. I think that's like pretty much the opposite because high able would lead to low guidance. We can mark that one out. So suppression of able reduces the severity of abnormal axonal guidance in PTP mutants. So if we're talking about PTP mutants, these guys, oops, not what I wanted to do. If these guys are gone, you can potentially help to alleviate some of the issues by also suppressing able, just so that you can restore some of that normal axonal guidance. So instead of PTP reducing able, you can artificially reduce able, and that's what would lead to reduced severity of abnormal axonal guidance. So that would make potential sense there. Does that line of logic make sense?
But when, oh, I see. Yeah.
Because this is basically saying PTP lowers able. If we don't have PTP, that's bad.
So,
But
yeah,
if we lower able on its own, that can help us.
right.
PTP lowers able. So instead, we just suppress able, and that helps us to recover some of our normal axonal guidance. And then we should know that we want low able, so we don't want B. And then we want low able, so it's not going to increase the severity, it's going to decrease the severity. Make sense?
Mm-hmm.
Awesome. Yeah, basically a direction. Oh, they drew it out. How nice. PTP, less able, normal. Without PTP, it's getting abnormal. But if we also can just find a way to repress able, we can still keep this process normal. So even though PTP isn't doing its job, if we target able, then we have more normal axonal guidance.
Wait,
Cool.
hold on. So with, oh goodness. Okay, wait. So without able, but with PTP, we would have normal. But if we have too much ABL,
So that's this first case.
yeah. But if we have too much ABL, inactive yields, PTP is inactive, which yields abnormal. But then if we have both of them not present, normal and abnormal, why would it be
So it's
restored?
abnormal. So normally able is low, and that's what's good. Low is good. When we inhibit this or have a mutation against PTP, that's bad. It's high. Able is high, and it's abnormal. So to make up for the loss of PTP, we can also just target able to lead to low able to restore our good outcome.
It's like the lack off
So basically,
glucose
yeah,
and lactose.
sort of. Basically just saying, this is more thinking of like if you've knocked something out, now that's bad, but you can also like help to make it good again by just like targeting its target for it, right? So in this case, instead of PTP lowering able, this is like saying that we as scientists lower able either way, you're getting rid of able, you're lowering able, and that's going to lead to the good outcome of axonal guidance. Does that make sense?
Yes.
Cool. So basically, it doesn't matter what's going on with PTP upstream because we've targeted able downstream.
Yeah.
So you're still able to have normal axonal guidance as long as you're able to target able.
Mm-hmm.
Awesome. Okay. Yeah. First circumstance is the same as the third circumstance because in both you're decreasing able. Okay. I think that was it for this guy. I mean, most of these weren't passage related. They were a lot more like Punnett square related, or like the slight language that they got that was related to like autosomal versus sex linked. But overall, I think
That passage,
kind of
I
makes
would
sense,
say it
right?
was solid.
And this is a good test.
Aside from that little passage, it was fine.
51. Okay. Okay. So now we have this passage about trait theorists. All people have certain traits. We have two studies. This is like the classic psych setup, and I know that one of the questions you got wrong was related to like comparing study 1 versus study 2. Okay, participants were asked to select one personality trait they wished to increase. They measured the baseline, and then the experimental group developed their own interventions, and the control group did not change anything. And then took the same survey to see what happened. Results were calculated by subtracting pretest. Okay, yeah. Basically, they're seeing what changed in between control versus experimental for people who were trying to change a trait. And they showed significantly greater change for experimental participants for this one particular trait of emotional stability. Okay. Study 2, the same basic methodology was used on a new cohort with several adjustments. Licensed therapists trained participants and practices to redirect thought practices, blah, blah, blah. Personality trait expression scores were determined by assessments made by researchers. Significantly greater increase in all 5 personality trait expression scores. Only difference we see subject or study 1 is very like self-design, participant design. Study 2 is a lot more led by like professional intervention. But in both cases were measuring traits before and after intervention and or no intervention.
Yeah, the thing that was weird with this was like it was taking one of the traits or 2 of the traits and then assuming a pattern with the others. And I just didn't understand that line of thought or correlation, which screwed me up with the questions. Because it was like saying that which one they picked out of these that they wanted to change, and then they did that self-report, and then the licensed therapists are the ones saying, well, this is what we're gonna test, and then all of a sudden it's more of, yeah, there is no significant difference with the others, even though I don't know. It's just, I feel like it's misleading in my opinion, but
yeah, so the traits they have: openness to experience, conscientiousness, extroversion, agreeableness, and emotional stability. So like basically, I think what they were saying is like people just picked one of those and saw if they could improve it. And then in the first experiment, the only one that was able to be improved was emotional stability. For all the rest of them, none of them improved even when they were trying to improve them. So like study 1 is basically saying like emotional stability is the only thing that you're able to improve. And then in study 2, they did the same idea, but they were able to show improvement for any of these. Basically, so like if they picked openness to experience, they were able to see improvement. If they picked conscientiousness, they were able to see improvement. with professional intervention. So I guess the idea could be like traits can be improved. Maybe you need professional intervention for some of those, but like in this one, you showed that this trait was able to be improved. And then in the second study, you showed that all of these traits are able to be improved with intervention plans.
Okay.
Okay. A trait theorist would most likely challenge the study findings described in the passage by asserting that researchers failed to assess, yeah, I like that, whether they were maintained following the period of intervention. I think that's really the only one related to the actual study. The rest of these are fairly unrelated to the study. Does that make sense? A researcher interested in whether there's a correlation between the personality trait of neuroticism and somatic symptom disorder is most likely to assess if anxiety about bodily symptoms increase as emotionally yeah, that's basically just asking you what is somatic symptom disorder and then connect it to neuroticism.
Mm-hmm.
Okay. When comparing the research designs used for study 1 and study 2, which of the following statements is not true? Okay. We'll just go like answer choice by answer choice here.
Mm-hmm.
A, post-test scores in study 2 are more vulnerable to experimenter bias. I mean, there would be nothing to indicate that. I think that's probably why you eliminated A.
Yeah.
There's no real difference in experimenter level of involvement here versus there.
Nope.
C, the design used for study 2 is more labor intensive and more expensive to implement.
That's true. They have licensed therapists. Like,
yeah,
they hire
it's
people.
definitely requires more infrastructure.
Yeah.
Oh, statements is not true. Ah, okay. This one was true because experimenter bias was that they were the ones evaluating.
Yeah. Rather than social desirability bias, like for study 1.
Mm-hmm. Okay. Then we're left between B and D. So let's look at D because you picked that. Post-test scores from study 1 are more vulnerable to subjective interpretation. I guess was the reasoning here in that it was like, I don't know, kind of self-driven.
It was more of what would be the counterbalancing measures? That's what I was trying to differentiate between. Is it more of an emotional, like connecting worth from the post scores, or is it more of like these other, like I didn't understand the counterbalancing part of it aside from it's the same self-report survey.
Mm-hmm.
But
So the best way to describe our counterbalancing is basically like that both the experimental and the control group, that you switch around your like experimental versus control. So it's like to say if you did 2 tests, one was to see like how much you wanted to eat a cupcake and one was to see how much you wanted to eat a spider. If you just did like spider then cupcake, people might want to eat the cupcake less or more because of the spider. So you can't control, you might be affecting the second part of that based on the first part, if that makes sense. So if you offer someone a spider, they say no, ew. Then you offer someone a cupcake, they might really want the cupcake. And so in order to control for that, you need to offer people spider then cupcake or cupcake then spider, so that you can kind of differentiate that the order you did things in is not actually contributing to something here. If people are being influenced by like doing one thing first or another thing first, that's potentially affecting your results. So counterbalancing in this case would be, first, you have someone try to change one trait and then you have them not try to change another trait. And then for the control group, you have someone not try to change one trait and then try to change another trait, just to control for kind of the order in which things are going and happening. And that's what kind of counterbalancing is.
So it's like
Is
choosing
that,
the order of the dependent variable.
yes. Choosing the order of the independent variable.
Oh, it's the independent. Okay.
Yeah. Okay. So this, yeah, they didn't really write that out. A to B, B to A. So in this case, what like you basically want to kind of control for the process. And so what you can do is basically just switch around the order in which things happen. So oftentimes what you'll see happen is you have like one group is control, one group is experimental, and then in the second half of the study they switch. And so that just helps you control and make sure that like within groups and across groups you're getting the same results. So it just provides another measure of control, because like you don't necessarily know if maybe it was just related to like someone thinking they could change something and not necessarily the process. I don't know. You just want to control for all variables. So normally you'll kind of see like swapping your experimental and control groups, and that's what counterbalancing will look like.
Okay. But is it safe to say that it's a validity thing that is actually what you're supposed to, or is that more specific?
Yeah, I think it could be a validity thing. I think for this kind of experiment, it's like less of a concern. Like I feel like you don't need to do it as much, because it's like you have experimental versus control. With validity, it's really a big concern when you're doing like two different tests. So you've got to do like test A, then test B, and test B, then test A. So like it's much more of a concern for like validity of your results in kind of those multi-part experiments. But it's still like, at least in the context it's used, you can see that like there was no counterbalancing in either. So there's no difference between study one versus study two.
So then what does the subjective interpretation play in for study one?
The way I was thinking was, I mean, they're doing like a self-report survey. And
Because
then in this case, they're doing like assessment by researchers. Honestly, I think that like each of these is pretty subjective. Like, I don't know if one's necessarily more subjective than another, because this is like self-report versus researcher report. Um, I think self-report is a lot more subjective than researcher report.
sure.
Obviously, we had the question of like experimental bias or experimenter bias. So that's more of a concern in study two, because the experimenters are evaluating. But in study one, subjective interpretation would be like if you ask someone to improve something and then they take a survey about whether or not they improved it, like they're probably going to be a little bit more biased towards
Sure.
like themselves. So that's what's opening up like self-reports are subjective interpretations. So that's why study one is more subjective. And then just in terms of like pure factual, there's no counterbalancing in either. So it makes sense that B is not true.
Is this also in comparison with objective too? Like, is that kind of the same line of thought? I know like objective is non-biased, but is it kind of like along those lines? Because I know there was a question about it on the exam that I took, this recent one about subjectivity versus objectivity. So is it like along those lines too?
I mean, two could be more objective, but still is somewhat like both are somewhat subjective. Because
Right.
in either case, you're having like evaluating someone's traits, like that's always going to be very subjective. Self-report is always more subjective because people are very biased about themselves, but also like experimenter bias is a thing. So if you were trying to get truly objective results, that's usually going to be like a response time test, something that you can like quantify with numbers.
Okay.
That's done in like a very like formulaic kind of way. That's, you know, a thing that's evaluated based on like response time or answer to a question, like something that's going to be like measurable and not necessarily up to like if the researcher thinks this or that or if the person thinks
I
this or that.
understand. Okay.
So what it says down here, you just scroll, is that because there was only one type of intervention used, their counterbalancing measures were not required. So that was basically saying like, you know, experimental group could be experimental group, control group could be control group. So it's only really necessary kind of if you have, because mine was just like A to B and B to A. In this case, there only ever was an A. So it was just like A versus nothing. So don't necessarily need counterbalancing in this case. It's going to be more relevant in terms of when you have two different tests, kind of like I described with the spider and the cupcake.
Okay.
Make sense?
Yes.
Okay. The amount of change for the control and experimental groups did not actually differ. If the amount of change did not actually differ, which of the following measurement errors would most likely result in a false positive? So how would we make an error that makes us think the experimental group increased more than the control group? Okay. Okay, so we have A, the experimental group participants in study 1 overestimated their pretest scores. So we know in this case, the scores, it said the improvement is post minus pre, right? Post minus pre. So I think that basically we're looking at like how much they improved. Does that make sense? If someone is overestimating like where they start at, I think that would actually make this improvement seem smaller and would probably make, if the improvement for the experimental group is smaller, then it's not going to be increased more than the control group, if that makes sense. Because control group is pretty much going to be 0, right? They're not going to change from the post-test to the pretest to the post-test. If someone's overestimating their pretest score, that's going to bring this number closer and closer to 0 because it'll negate all of that improvement. So I think A would have like the opposite effect, if that makes sense.
We're trying to find, I think this was more, I was confused on what I was trying to find because I didn't know, like it was saying which one would be given a false positive. So experimental group increased more, but yet the control experiment change was like nothing. So I couldn't tell if it was like pre-post for control and then pre-post for experiment, or pre was control and post was experiment. I don't know. It was just that line of thought.
I get it. No, I think I get what you're saying. So does it make sense if I'm testing, let's say, my emotional stability? I rate myself a 5 before we start. So that's the pre-test out of 10. Then I go through this intervention. I'm like, wow, I'm so much better. I now give myself a 7 in emotional stability. That would mean I improved by 2. Makes sense? Control group, if they started at a 5, they're going to end at a 5, they're going to get 0 improvement. So this is saying if the experimental group actually increased, we'd expect to see a bigger number here for improvement.
Right.
But if there's some kind of error, we could see this number as bigger even though it's actually not. So we're asking basically what could happen that would lead this number to be bigger than 0 even though it's not. Question A is saying if you're overestimating your pre-test score. So let's say I'm actually a 5 I'm like, if, I'm pretty good. I'm a 6. And then at the end, I evaluate myself and I'm still a 7. Now I've had a 1-point difference. So now I'm actually closer to 0. So this difference has become a lot smaller. If
I
I'm
see.
so overconfident and I overestimate my own score, maybe it's a 7 and I don't actually see any improvement because I was faking it in the beginning.
Mm-hmm.
So if you're increasing your pretest score, that's going to bring experimental and control closer because it's
Okay.
going to basically erase the improvement that you see in the study, in the experimental group, if that makes sense.
Okay.
So I'd eliminate A. And then in B, it says all participants in study 1 underestimated their pretest scores by the same amount. I think that's what you'd crossed off, but that would, kind of negate the difference because
Agreed. Yeah.
if if I'm saying this is 3 and they're saying this is 3, they've kind of decreased by the same magnitude.
Right.
Okay. Researchers in study 2 underestimated the pretest scores for the experimental group participants. So this is now where instead of me evaluating
That's
myself,
the opposite.
researcher evaluating me. And so this is saying, imagine that when I start off, I'm a 5. The researchers are like, she's probably a 3. Now this improvement looks much bigger and much better. I went from 3 to 7. I improved by 4 instead of going from 5 to 7 and improving by 2. If they're underestimating the starting point, then the ending point when they've improved will look so much better.
Got it.
This is basically saying that, like, if you're just underestimating the experimental group, you'll see improvement even if nothing's changed.
Mm-hmm.
If everyone's a 5 at the beginning and the end, everyone's 0.
Right.
But if they call you a 3 at the beginning and then give you a 5 at the end, you've improved even though nothing's changed. Does that make sense?
Yeah. Then D
Slightly
is
confusing.
true.
But
No, it makes sense. Also, it makes sense why study 1 wouldn't even like be fair for this question either, because it's all self rather than like someone giving you or like analyzing you, if that makes sense.
mhm. I mean, it could definitely be self-related. Like someone could overestimate their own improvement, and that would be a way that you would get a false positive. But that would be more like self-bias, like subjective bias, than like
Right.
measurement error.
Right. Yeah. And then D doesn't make sense because you can't do it by the same amount either.
Yeah.
Okay.
Basically, if you underestimate where you start from, any improvement will look bigger and better, and
Yeah.
that will give you results that look significant, even if they're not.
Mm-hmm.
Okay. Psychoanalytic theory would most likely suggest that experimental group participants who are able to alter their personality traits have. So this should pretty much directly point you to Freud, obviously, because
Yep.
you see superego, id, whatever.
Yep.
So they're saying that if people are able to change their personality, what traits do they likely have? So we're looking for something that's going to be like, I guess, capable of executing change, capable of like actually changing. So probably going to be dealing with the much more like conscious like side of things is going to be making actual choices. So it does look like, yeah, you were between A and C, right? Because you're related to ego, super ego, like things that are more conscious decisions.
Okay. I didn't even think of that. I thought it was more of like, because I thought because you're changing the personality trait, maybe you want to perfect it or like become better than what you were originally. So that's why I thought of super ego rather than, well, what are you trying to counterbalance between your impulsivity and perfection, but not at that like the conscious and unconscious level or subconscious,
Mm-hmm.
not subconscious, preconscious.
Yeah, that's fair. I mean, I think that, yeah, I think you could argue then for either ego or super ego in terms of like something that's going to be executing improvement. I don't know. Yeah, I definitely defaulted straight to like the conscious side of things, but yeah, you can also just eliminate super ego and id based off of like neither of or sorry, you can eliminate id based off of the fact that it's not going to really be related to, I guess, improvement
Yeah.
in this way.
If anything, it's the opposite. I mean, it's not neutral, that it's always improvement. It could also be harmful,
Hmm.
actually.
Yeah, because I think, yeah. Okay. So then if you just go based on the like conscious angle of like conscious change, that's where it would point you to ego because that's the only one that's like more on the conscious side. Um, I think the super ego, I see where you came from there and what you were talking about with like the perfection. Um, I guess strive to like be better at something. The only thing I didn't necessarily like was like overcome defense
That's the part
mechanisms.
that was like, that was like the iffy part because there's nothing really about, oh, I guess that too, defense mechanisms are unconscious strategies. Oh, I didn't even think about. Okay.
Okay.
All right. Well, that's why that's wrong then. So I guess not. It's just the moral line. Okay, that's fine.
Okay. Okay, so that wouldn't even align with what the super ego does. Okay. So I guess it was like if you're on this graph and you drew defense mechanisms would have to be up here. So the super ego couldn't even overcome defense mechanisms because it's all the way down here.
Yeah.
And then id, we said the pleasure principle is a lot less related. And we also know that ids don't relate to
No,
reality
no, that's the
and ids don't really relate to rationality because they're also so like definitely not the id in either of these cases. um it's like we could see it being the super ego, but it doesn't really relate to defense mechanisms, so it's probably going to be more related to like conscious change that someone is pushing to do, which I think can fit with like ego reality principle.
Mm-hmm.
Mm-hmm. Cool. I did not realize that defense mechanisms were associated with the ego, but that's good place to throw that on a flashcard or something.
Yep.
Awesome. Okay. Cognitive behavioral therapist in study 2 uses principles of classical and operant conditioning to treat patients with extreme introversion and social anxiety. The methodologies employed by the therapist would least likely include. Okay. So we're basically looking at like classical and operant conditioning. So maybe something that is not classical or operant conditioning would be what they're asking about.
Basically.
Okay, we have A, repeatedly exposing the participant to extended period of social engagement. Probably going to be related. I don't know. I think of like fear, like the
Yeah.
word like fear conditioning or whatever, where they're like just exposing you to whatever, but they would potentially be exposing the participant and then rewarding them in some way to help them overcome that introversion or anxiety. And so that would probably be related to conditioning. We have commending the participants for attempting to engage in socially during free time. That would be a form of reward. So that would be how they're like classically conditioning someone to like respond to a reward, or operantly conditioning someone.
Yep.
Requiring the participant to eat favorite foods only when in the presence of company. The way I look at this is favorite foods is the reward, and it's only when they're in the presence of company. So it would be like, oh, you get a reward when you engage in social behaviors. That could be kind of viewed as conditioning, and that like if you do the undesirable behavior, you get the reward.
I thought of it as positive punishment.
Positive punishment. So positive punishment, I guess, would be the presence of company.
Yeah. Well, I guess punishment is taking away. So, or I don't know, something along, what was it? I don't know, it wasn't negative reinforcement either. Well, no, that doesn't make
Because
sense. I think it was like underline, like along those lines. I don't know the train of thought, what it was at the time, but
because if you think about punishment is trying to like avoid a behavior. So like you're training someone to like not do something. So positive punishment is when you like give someone a shock if they do a behavior. That would be like you're positive, you're giving someone something, but it's a punishment because you're trying to like teach
avoid
them
that.
to avoid a behavior. So positive punishment would be a shock if you do something you're not supposed to do. Negative punishment would be if you do something you're not supposed to do, I take away your food.
Mhm.
So in this case, we don't want punishment because we are wanting to enforce the behavior of being in company. So in that case, we're looking at reinforcement because we're trying to teach to do a behavior. We want the person to want to do the behavior. So we can have positive reinforcement. I give you a cookie if you do what I say, or negative reinforcement. I stop shocking you if you do what I say. So I would think of punishment and reinforcement in terms of like, what's the desired outcome? Are they trying to teach someone to avoid or do something? And then positive or negative in, are they like giving them something, whether that's good or bad, or taking away something, whether that's good or bad?
Mhm.
So in this case, we want the person to be in the presence of company. So that would be a reinforcement. And in this case, we're giving them a reward when they do what we want them to do. So that's positive reinforcement, which is operant conditioning.
Okay.
Either way, punishment, reinforcement, all of those things are all like
Okay.
related to operant conditioning. So I think that would be something like related to what the therapist would do.
Mhm.
And then D, I think, is just the answer because it's unrelated to conditioning. It's just at like observational learning.
Okay. Okay.
Make
Sounds good.
sense?
Yeah.
Uh-huh. Yeah. I feel like the positive and negative sides of things can always get kind of complicated, but I just like to break it down to first punishment versus reinforcement and then positive versus negative, and then I think it makes sense from there.
Mhm.
Okay. Do. Okay, that was that one. Does that make more sense?
Yes.
Cool. Okay. Yeah, these are all pretty solid, aside from those like one or two paragraphs, but even those were like, you know, just a couple fill in the blanks, and hopefully those will make more sense in the future. And hopefully that like what's it called? The I don't know, shortcuts and stuff will be more helpful.
Mhm.
Okay. The last thing I wanted to do quickly, I spent a lot of time talking today, is to just glance over the cars from this most recent test. And just kind of look at, let's see, what's this ROT?
Ran out of time.
Ah, okay. Stupid passage, silly mistakes. Okay. Let's just, I think, yeah, this might be best to do like somewhat on your own in terms of like trying to map out what would be the best
Yeah.
way to like diagram it out. Um, if you want to just kind of do that with the passage and then send it to me, I can also do that same passage and like kind of show you what I did.
Mhm.
Like if we want to just both pick a passage and then kind of compare how we each mapped that out, I think that could be helpful. And then basically just kind of going through, now that you've obviously seen what are the right answers and what is the right thing, and seeing if that kind of map of the passage would then make more sense when you're going towards looking at the questions.
Mm-hmm.
The other thing that I've been like starting to do, let me see if I can find a good question stem. Okay, maybe I'll have to just find this for next time, but I was just going to show you like an example of, I've been like finding ways to kind of break down, here we go. The question stems that I think could be helpful. So just to kind of exemplify what that would look like. So we have like this kind of like assumption coming into play here in red. Assume that in a particular state, teachers at all grade levels emphasize hands-on projects and personal interactions with individual students. And then the passage suggests, and then we have two, the passage suggestion about class size reductions. Just kind of being able to identify the different portions of the question stem that are being referenced. So like one question that I saw a good example of this was like, here's a new piece of information, how would this contrast with this assumption? And so just kind of differentiating out like, okay, this portion in red here, this is my first new piece of information. Here's my new assumption, here's my passage assumption. How do these two relate? Like, what is the relationship between these two, kind of thing? Like, if you can just kind of break down that question stem and understand like, okay, they're asking me like, does this support or deny that? They're asking me, how does this one affect that one? Basically, just being able to break out the different parts of the question stem can be very helpful.
Okay.
So potentially if like, once you've gotten that all mapped out, it might be helpful to just show like, okay, here's my assumption. This is all question stem based. Here's my passage. This is where I've got to go back, reference the passage, pull some evidence. And then now the question's asking me, how do these two relate to each other? How does one influence the other? Just to kind of differentiate out, what are you actually getting from the passage versus what are you supposed to be kind of like coming up with on your own?
The flow chart will help with that too. So then we'd just be like filling in as I go through questions, just be like, this is a new information, how does it affect this? And then basically see that cascade.
Yeah, almost like an extension of, oh, I like this one, like an extension of the flow chart, in that you have like, oh, like this thing, that thing, whatever, whatever. And now this question stem is like new idea number one. Where does this fit in to the flow chart? Basically, is the question. So your answer is going to be connecting to one of these points on your flow chart.
Yeah.
In this case, it's saying, which of the following findings? So we're looking for, the first thing is just like an answer choice. Would most weaken the author's explanation of the observed effects of class size reduction? So the way I would kind of draw this out is, so we're looking for, firstly, what is the author's explanation of class size reduction? Then we're looking, secondly, for one of the answer choices. And then we're looking, thirdly, for something that weakens. So the way I would go about it is first identifying the author's explanation, then going through each of those answer choices, substituting that in here and seeing which one makes the most sense in terms of weakening the author's explanation. Just kind of being able to pull out those different parts. And then you can also, when you have that in your flow chart, this is going to be pointing towards this arrow, but we now want something that's like going to disagree with this arrow, something that's going to disagree with this portion of the flow chart. If that makes sense. Okay. Just wanted to like provide that as a way to like kind of break down the question stem that could fit into the flowchart approach, potentially. Okay. Cool. Well, okay. I know we did through a lot of the UWorld stuff today. Didn't necessarily get to any of the full-length stuff. Did you want to have a meeting again soon to go over any of the like full-length specific stuff, or what was your like big takeaways from that?
Where's my hodgepodge? Oh, I think honestly, I mean, I addressed the little things from chem, basically scientific notation, flowchart, and talked about that as well for BB. Honestly, I think it was fine. I think it's just more of being more and more exposed to just through practice, honestly. Nothing really in particular with like specifics. Because overall it felt fine, just more of a review and apply to next time kind of situation. And then figure out what needs to change with cars, if that can be improved.
Yeah, totally fair. And then are you planning to take a full length this weekend, right? Full length 4?
We said this weekend was more of like another hodgepodge part 2, kind of. I don't know what that consisted of. I think it was more of just, I don't know if it was actually like treating it as a full length, but definitely like going through past section tests or past, and going through.
I think so. I would do, if you think this is possible, like full length for this weekend, and then do that kind of like more relaxed one next weekend, because then the weekend after that is another full length. So then it would give you like sort of a weekend off in between. Unless you think that you're like, I can just power through. Because then you could do like real full length, slightly less obnoxious weekend, and then real full length again.
Honestly, I was in the mindset of this weekend being more chill and then next weekend having that back to back. So
totally fair.
I don't know. I just feel like we're already midweek or past midweek. So changing that mindset.
No, I don't change it. Oh, yeah. So what I would do, I know you've redone a couple sections. You still have AMC, well, the passage or the sections that you didn't finish from AMC, like that you didn't do from 1, 2, and 3, and the unscored. So I would just basically look over. This is the one we said it could be like not even like a full section's worth. Like, oh, I did really bad on unscored cars number 6 and AMC 3 number 7 kind of thing. So just piece together what you want to do, and basically the point is to just be doing AMC content, which you will be doing.
Yeah,
I would go through one of those.
No, I was saying that's definitely doable. I'll just write it out what needs to be reviewed.
Awesome. Yep. I would yeah, just do it. Do what you think you can do. Get it done. Feels good. I would do a lot of the cars passages again from like the 118 section, just to like, I don't know, see how they feel now. Maybe they still feel terrible. That's fine. I just want to see like if they feel any better. Maybe kind of as you're messing around with this new mapping strategy. Yeah, I'll be around. Just text me and let me know kind of what you're thinking, how it's going, and if you mess around and it still doesn't necessarily feel like it might work, then we can kind of look at that next week and try to think of some alternate approaches.
Yeah, sounds good. I'll definitely fiddle around with it for the next few days and see what works best.
Cool. Awesome. Let's then plan to meet sometime next week. You can just kind of let me know how it goes this weekend, and then we'll judge from there.
Sounds good. I will do that.
Awesome. I will see you soon then.
Sounds good. Safe travels.
Thank you. All right. Bye-bye.
Bye.